Okay, everybody, welcome back from lunch. Let's get started so we can keep on time. So I'm Helmut Zarbel, I'm a member of the committee, so I'll be chairing this second session, which is the next topic out of the three that we picked. A very exciting area of copy number variation and chromothripsis, if I can say that correctly. And without further ado, I'll introduce our first speaker, James Lupski, who will put it in mechanistic and evolutionary context for us, and then we'll move on to the environment, I guess.
Thank you. Thanks very much, and thanks for the opportunity to share some of our lab and department's data with our colleagues here at the National Academies. I thought I would divide the talk into a couple different topic sessions, give a little background of the thinking behind copy number variation and gene dosage, talk about copy number variation mechanisms, in particular the newest, not NAHR, but how it seems to happen by ectopic synapses. The third, again, a newer mechanism for triplication, where inversions then lead to creating brand new genes by flipping the DNA over and reading the other strand. The fourth topic, just touch briefly on chromothripsis, because this is all very, very new to us, and we're all still trying to figure out how it's happening and to what extent are these massive genomic changes taking place, particularly in cancers. And then finally, a little bit about evolution, and to lead into the next talks about environmental mutagenesis. So we had a great talk this morning from Thea, and she nicely introduced interpersonal genome variation, which in the human as a model organism, Homo sapiens, I tend to think of as germline genomic variation, and my petri dish is the world. But chromothripsis is really somatic intercellular variation, but we see the similar type of events happening in the germline, or at least I'll try to convince you that that's so. So as I scanned through lots of literature and textbooks, I tried to find out when people started to think that dosage alone was really very important to a phenotype. And I found this paper from Cold Spring Harbor Labs from the 1920s when Al Blakesley was looking at the Datura, and it had 12 chromosomes, and he could find a plant that had a different phenotype depending on the trisomy you had. So each individual trisomy led to a different plant phenotype, and really he put forward that just dosage alone and chromosome copy number could be important to presentation of phenotypic expression. We jump ahead a couple of decades, and Calvin Bridges is working in the Fly group, working very, very hard, and he gets a new technique, just like we get arrays in exome sequencing. He gets a new technique, and this is polycene chromosomes. And as he starts looking around the genome at different phenotypes, he finds that the bar locus has a duplication. And it shows you on the left the phenotypes that you see in the males and the females, and what you see when you get a slit eye or bar. Homozygosity gives you this, which you can also get for triplication. And he found triplications occasionally, but didn't know if they were originating from the normal copy or from the duplicated copy. And I'm going to show you how it happens both ways in humans. But interesting to go back to his original paper, because he thought very hard about his data. And he was at a time when position effect was everything in genetics, but he specifically states in his discussion that the respective shares attributable in the total effect to the phenotype based on whether it's gene balance change or dosage or a position effect change seems to be at present a matter of taste. And taste in those days was position effect. So some of these things got buried in the literature for some time. In humans, a series of a couple of papers came out in the 80s mostly. Doug Higgs at Oxford, Jeremy Nathans, then working at Stanford as an MD PhD student, Rick Lifton in Utah before he went back to Yale, And all of these cases found duplications, but the rearrangement disrupted the very gene involved in the process. So the thinking was still that there was some kind of gene interruption, and that was being responsible for the phenotypic expression. I dug out this paper, which I really like, because the authors nailed it. And this is a paper from a French group working here at the NIH with Colin Gadjicek. And this is 10 years after he got his Nobel Prize, 10 years before he was incarcerated for pedophilia. And they did a great experiment. So they found all Alzheimer's disease families that had increased copy number compared to the normal control, and it was identical to what they saw in Down syndrome, and the APP gene had just been mapped to chromosome 21. This was very, very exciting. Here's all the controls, normal individuals, other AD families that were not inherited, beautiful control. Down syndrome clinically, but normal chromosome number. These have subsequently been shown to be submicroscopic duplications of the Down syndrome region. I love this experiment. I love the data when I look back on it, but these guys weren't working in the Alzheimer's field. So the gurus of Alzheimer's proved them wrong. And Science took 2 papers, twice the number of pages, with negative data because the gurus pushed that this cannot be right. Less than 10 patients. Really remarkable to me. Of course, 20 years would go by. First, that would stymie a lot of work in duplication and dosage field. 20 years would go by, but the very same population, the French individuals, were shown by multiple methods to have duplication associating with autosomal dominant Alzheimer's disease. And I hope we all can learn a lesson from this social science experiment. Now, of course, now we've known for decades that patients with trisomy 21 have early onset Alzheimer's disease. We never knew why, but they did. It was just a clinical phenomena. We saw that duplication could cause it. GWAS studies have shown some signals for non-coding upstream single nucleotide variation, and there are rare families with point mutations in the coding region of APP. Likewise, for Parkinson's, we can have non-coding variation, or we can have point mutations in the alpha-synuclein gene, and both duplications and triplications have been described. So we start to now, rather than dichotomize common disease, Mendelian disorders, chromosome syndromes, we can look at it all as human genome variation and try to really get at heuristic models for what are the underlying genetic architecture of different diseases. And this is becoming a very hot topic now, that we can sequence the entire genome. We can sequence it in your mom and dad, and we can see exactly what happened in that generation, the whole genome-wide, not just at a single locus. So, generally speaking, when it comes to structural changes that can cause copy number variation, 4 different mechanisms have been elucidated. I'm not going to talk about L1 retrotransposition because I never quite understood it, and I won't do anything like John Moran can do, and we'll hear from him later. I'm not going to talk about non-homologous end joining. It's well established. I will talk about these 2 mechanisms, the ectopic recombination, which has been called non-allelic homologous recombination, and the replicative mechanisms of fork stalling template switching, later further delineated into microhomology-mediated break-induced replication. So, at the same time, all of the investigators were proving the Alzheimer's group from NIH wrong. We also saw duplication at the Charcot-Marie-Tooth disease locus and felt we had to prove this multiple different ways, use 6 different molecular methods independently. Christine von Brockhoven's group in Antwerp reported the same thing. We were back-to-back speakers at the same meeting, had no idea the other had figured it out. We went on to actually show it truly was gene dosage and that the mechanism was a non-allelic homologous recombination where flanking genomic repeat sequences lead to a duplication and change the dosage of a critical dosage sensitive gene that predicts a reciprocal deletion event. The disease locus was predicted, but took a couple of years for Phil Chance's group to demonstrate it clinically linked to hereditary neuropathy with liability to pressure palsies. Remarkably, although point mutations in over 50 genes can cause Charcot-Marie tooth disease, 70% of all patients have this duplication described over 20 years ago. Great clinical diagnostic test. Even more remarkable, if there's no family history of disease, you have sporadic Charcot-Marie tooth disease, you have up to a 90% chance of finding this duplication because the de novo rates of CNVs are orders of magnitude greater than point mutations, at least in the humans where we study. Now, the mechanism's a little bit complicated. Try to explain it to a parent during a genetic counseling session. And that's why I need people like Lori Pataki, who's a terrific clinical geneticist, who translates this information. You can think of it as here being the haploid genome. In white are the repeat sequences flanking the dosage-sensitive region in orange. I like to swim in the ocean, but I do not like to swim in the pool. With a deletion, I like to swim in the pool. With a duplication, I like to swim in the ocean, but I do not like to swim in the ocean, but I do not like to swim in the pool. Now, we haven't lost any information, but we've changed the content. Don't want to take the analogy too far, but most times deletion gives more severe disease than we see with duplications. So, studying these set of disorders, as well as chromosome microdeletion and duplication syndromes, we saw they were all happening the same way. So we put forward the concept of genomic disorders. These would be diseases due to rearrangements, not Watson-Crick base pair changes. And furthermore, that the architecture, as we showed for NAHR, would incite instability. So the focus for 20 years is figure out how it happens mechanistically. Then, as we move in on the human genome, we can make predictions which will help guide our diagnostic tests for genome instability. Back to good old Frivoli and Calvin. He really looked carefully at his data and saw some of these triplications. He could get dupes to revert at a pretty high frequencies, a little harder with the triplications. We've started to find families where you actually have more severely affected members who have triplication that actually came from duplication in the previous generation. But at other loci, we've found you can go to triplication right from the normal copy if you have a recombination hotspot motif that can allow double crossovers to occur in a small space. And these are very, very exciting times as we think about triplications, and I'll get back to them in a little bit. But this mechanism has been shown to be the cause of recurrent deletion syndromes, recurrent duplication syndromes. And what I mean by that is the same rearrangement keeps happening recurrently over and over again in the population. Moreover, it depends on whether you do it interchromosomally, intrachromosomally, or intrachromatidally. The intrachromatid can only give you deletion. The mechanism favors deletion 2-to-1 at every locus where it's been looked at. You can also get isochromosomes. We showed it for ISO 17.6 in cancer somatic cells, but it's also been shown constitutionally in Y and X. And last year in genome research, we actually made a predictive map of recurrent translocations, and several have now been found based on this predictability map after you learn and understood the mechanism. So this mechanism looks important. Alec Jeffries has beautiful work now showing an evolutionary rheostat in different areas of the world. he can measure PRDM9 alleles and in the sperm of males find different frequencies of the CMT1A duplication by single sperm PCR. So this helps define a new discipline of medical genetics. And then by the time we had in the post-genomic era some good array technology, we could start looking at patients. And I'm happy to report, first of all, that's a group effort. This room, every individual there has met for 8 years, once a week. It includes 15 faculty members that have to come together. PhD mathematician who we hired in our genetics department, computational scientists, clinical geneticists, but we have now signed out 50,000 genomic arrays on patients and have a 20% hit rate. That is, 20% of 50,000 people have a diagnosis that before this approach, they never had one. What we learn? A locus can be subject to recurrent events, the NAHR mechanism, but non-recurrent events, which we didn't know the mechanism. All rearrangement mechanisms are possible at a locus, but you may favor one type because of the local architecture. Gains, losses, and complexity occur. These diseases are often sporadic because of de novo mutations, and the new mutation frequencies for CNV as measured by locus-specific rates are 100 to 10,000 times greater than point mutations at a locus. I just kind of want to take us through the definition of a genomic disorder. By the mid-90s, we had a mechanism. We found a deletion at this locus was actually observed by Ann Smith and Ellen McGinnis, the Smith-McGinnis syndrome. We found the mechanism. It predicted the reciprocal duplication. Took 3 years to find 7 patients and pulse field to show they had the same junction fragment. And now we define a new disease based on a predicted mechanism. Takes 7 years for Laurie to bring dozens of patients into the clinic and for 1 week do massive phenotyping so we can describe what the syndrome is. I didn't name the syndrome. That's what the reviewers named it. A couple months ago, another family gathering, the only second one. These families, the database has 300 patients and families from the U.S. alone in a disorder that was not even described until 2007. These are out there everywhere, and with the powerful technologies, we will start to diagnose them better. Let's now use the patients as our petri dish. They're the ones that are the purple colonies. Let's do the molecular analysis on them. Okay, so we do the molecular analysis. Here, 79 patients with duplication, 131 families with deletion. We see different modal distributions for the frequencies, and as might be expected, the recurrent ones occur at a greater rate. We can then look at the recurrent ones and try to understand the features which make them have different frequencies. The low copy repeats are similar in identity, 98.6%. So the question is, why is one more prevalent than the other? And it's totally related to the size. The correlation coefficient on the left, just size as a log frequency. On the right, factoring in distance. We don't get any pure correlation from the distance, but we get a little bit better by doing it by that ratio. This has led to a model, since synapsis happens by double-strand breaks, and the frequency of double-strand break will be greater with the longer homologous regions just by chance, that ectopic synapsis precedes crossing over. And this model, it's known that synapsis starts from the telomeres and moves in, so things that are closer to the centromere might be more likely to misalign and have an ectopic synapsis, like we see for the PTLS locus. I'm very proud of Peng Fei. He actually got the Kahneman Award, which he'll go receive that next month at the American Society. And his idea is consistent with the fact that we know non-allelic and allelic homologous recombination hotspots coincide. They use the same PRDM9 hotspot motifs, and really incredibly, in data which he is allowing me to show, it's not my lab. Shinohara sent me the slides, not even the submitted paper yet. If you actually knock out the synaptonemal complex in yeast, you completely abolish crossing over. So we design the model in the human petri dish. We test it on yeast. And this has been the procedure for several years. Triplications. So I'm going to focus now on the replicative mechanism. As we looked at the non-recurrent rearrangements, we thought we would just find a tandem duplication. It's not what we found. The majority of patients had snippets of DNA grabbed from several different pieces of the genome and put together. At every fusion, there was microhomology. This led to the concept of microhomology-mediated joining by priming DNA replication, and that this was a template-driven juxtaposition of DNA sequences separated by large genomic distances through a template switch mechanism. And Jenny Lee's the graduate student who came up with this. So the model has long-distance template switching. We saw it happen hundreds of kb and up to even a megabase away, tethered to the original fork, priming of replication via microhomology, template-driven juxtaposition of the discrete segments. Upon further reflection and additional data, it became apparent by Greg Ira's work in yeast that you actually process that 3' end to a much greater extent than all of us thought. That leaves a 3' end out there hanging. So microhomology-mediated break-induced replication is the concept of break-induced replication out of the Haber lab and Lorraine Symington's lab, with the idea that you process that 3' end, which is around and can prime, and that it uses some kind of low processive polymerase, so it comes off and on constantly. I just want to distinguish that if we think about DNA rearrangement, the Shapiro model gave us base pair duplications that flank the transposon upon insertion because it leaves free from the target a 3' end to prime through and replicate through the transposon. In this case, we use the microhomology to prime. So instead, what we see is microhomology that flanks the region that is brought together. Now, genomic studies, both locus-specific studies shown above and genome-wide studies, have showed remarkably that for breakpoint fusions, overwhelmingly what we see for normal copy number variants or pathologically associated ones is microhomology. And the latest data from 1,000 genomes, 10,000 breakpoints, 66% had microhomology. This mechanism drives what is happening in the human genome. Interestingly, the mechanism favors gains. So we saw NAHR favor loss 2-to-1. Here we see gain happens more often, duplication and triplication, and this is the major driver of the evolutionary process. From the MMBR model, you can get duplication, deletion, inversion, translocation, triplication, and as we heard from Thea this morning, rolling circle amplification, dihydrofolate reductase amplification, all comes out of this model. Now, when we see triplications, we see two mechanisms so far. One is exactly what Calvin saw. We saw a recurrent triplication. We see it in CMT. You go from dupes to trips. But at the STS locus, remarkably, 3 out of 61 families, 5% of all gains go to triplication. And here, the crossover occurs, as I had mentioned, there's a recombination hotspot. And in fact, the hotspot itself is a mini satellite. Mini satellites were defined by Alec Jeffreys, and this was the basis of human identity. And it was very exciting to see Alex when he saw this was happening here, and he started to study how it happens in the germline. The second type of triplication is unbelievably more complicated than we ever envisioned. Claudia, terrific postdoc from Brazil. She banged away at this problem for 5 years. It was published in Nature Genetics in November. We kept seeing, rather than the standard triplication, we got dupe, trip, dupe. And she spent years on breakpoints of hundreds of patients. And remarkably, although there's 4 transition states of copy number, there's only 2 junctions. And this happens because of the way the mechanism uses inverted repeats to flip around the genome and bring it together. Here's the mechanism. You can get the details, but basically it has an MMBIR step followed by a second step that one occurs in the inverted repeat sequence, so it uses homology for the MMBIR step. The second one occurs by microhomology. So it's like a classic break-induced replication with an MMBIR step together. We're seeing it around the genome. These are patients with different diseases due to triplication. Even the Charcot-Marie tooth disease locus, we occasionally see it go this way. This one here is the LIST1 locus. Deletion causes lissencephaly. Duplication causes a different disease with subtle brain malformation. But if we actually compare the MRIs in a blinded fashion, giving them to Jill Hunter, international UK expert in neuroradiology, she can pick out the triplications from the duplications. Moreover, when you do the triplication and flip the sequence around, it predicts a new gene is made, and Claudia can find those transcripts. So every time you make this mechanism, you create a new gene, completely new, because it's flipped the DNA over and it's reading the other strand. It's really quite remarkable. I thought I'd bring this up because of my audience. Not published, got rejected from the first journal we sent it to. But besides this complexity that happens at the breakpoint, we are seeing point mutations accumulate downstream. So this tells us that the polymerase in MMBIR not only has reduced processivity, falling off the helix, it also has low fidelity. And the point mutation rate is 1,000-fold greater than the normal polymerase. And remarkably, Anna Malikova has just showed this in yeast. And there's beautiful work that hopefully will be coming out in Nature soon between her group and Gregor Iris showing how it happens. Now, just because of the audience and what I heard this morning, I thought I'd throw in telomeric rearrangements because we all know and love Barbara and breakage fusion bridge. When I was an undergraduate at Cold Spring Harbor 34 years ago, it was Barbara who I spoke to about my career. And we can see at telomeres that this dupe inversion trip dupe occurs quite frequently. And I can't tell you if it occurs more frequently than breakage fusion bridge, but it's happening. And each telomere we look at it, when we see a triplication, this is what we see. Moreover, Now that we found the substrate sequences that stimulate it, this has come out this week in Human Mutation. We can look around the genome and find what regions will be susceptible and design our diagnostic arrays to catch it. In fact, for a regular inversion through NAHR, there's 942 genes that can be disrupted this way. And for the dupe trip inversion dupe, there's over 1,500 loci. Chromothripsis. My DNA world was stood upside down when this paper came out. Take the Cancer Genome Project, and I didn't know if anything was going to come out of it, but after only sequencing 800 genomes and comparing them to normal constitutional genomes, Mike Stratton, Peter Campbell, and others at Sanger saw this remarkable complexity that occurred chromosome-specific deletions, duplications, inversions, and really looked like they were one-off events. That entire process occurred once, and they had a lot of good arguments and data to support that. But more remarkably, in every cancer they looked at, 2 to 3% of the cell lines had this. In bone cancers, which are highly resistant to therapy, 25% of the cancers had this. It's since been shown that this is a marker to therapeutic response and to relapse in every cancer where it's been looked at. It's a single catastrophic event, not progressive rearrangement model. And multiple cancer genes are mutated in a single mutational event. So contrary to what we've all believed, single oncogene, single tumor suppressor gene, one mutational event hits 3, 4, 5 genes at once. Multigenic inheritance. We had already seen the event as constitutional event. That is, where neither parent had it and the child had it. In 10 of 12 of the patients, they were referred for global developmental delay. 4 out of 12 had intellectual disability and behavioral problems. All of them had some degree of developmental delay. Here's just an example of massive changes with the parental array shown next to it to show these are all de novo, in contradistinction to what they saw in Sanger, where they did different methodology. And remember, with genomic sequencing, you filter through a suboptimal haploid reference sequence. So much of the good stuff that I like, you're throwing out because it doesn't match. The computer can't match it to the reference sequence. Suffice to say, we also saw multiple triplications, which are very hard to explain by a non-homologous end joining. Rick Wilson did a beautiful job to explain that Sanger saw the phenomena, we saw the phenomena, we interpret them different ways. And we think our data suggests that the replicative mechanism is important here also. So we see 3 distinct types of highly complex human genomic rearrangements. The one-off event, so-called chromothripsis. Then we've seen events where we have them happening all over the genome and not in either parent. And then we see abnormal segregation, but we've seen this mostly in CVS, that at amnio we don't see it, or their early terminations. This is a multiple de novo rearrangement event. There are 9 megabase sized duplications or a triplication in this patient all over the genome. Mom and dad don't have any of them. And why do we see the patient? Because one of the duplication catches the dosage-sensitive PMP22 gene, and they present with Charcot-Marie Tooth disease, but with developmental delay. Likewise, here's another event. There's 12 megabase size duplications and triplications. Neither parent has it. CNV and evolution in my last 2 minutes. Actually, I have 3 minutes and 30 seconds. I'm doing better than I thought. The DNA replication mechanism, FOSS, MMBI, it causes dupes, trips, complex rearrangements. It creates entirely novel genes by inverting DNA. It may be a major mechanism for duplication CNV and thus a major driver of Ono's gene duplication divergence evolutionary hypothesis. And remarkably, by switching the template, grabbing an exon and coming back, you can shuffle exons all over the genome. And we see it happening a lot, as well as seeing single exon dropouts that are being missed by exome sequencing and clinical resequencing of genes. They're happening quite frequently. So the last time I was in this building was September 2008, when there was a meeting celebrating Darwin's 200th birthday. And for my celebration, it also happened to be my 50th birthday, and my family brought me to the Galapagos the month before. And these are pictures taken by my kids on the 11 different islands that we visited in the Galapagos, and then here shown sitting on the equator on the mainland in Ecuador with mom trying to keep the two sides of the family in check. Now, Leif Anderson has published remarkable papers over the last 5 or 6 years where he's used animal evolution and domestication of animals, which we all love and are near and dear to our heart, to map the genetic changes causing these traits we've put under tremendous selection. Every one he's found so far is a CNV. These are rapid ways to evolve phenotypes. And I'm going to close basically on this example. At that very meeting I was at in September 2008 here in this building, put forward by Bernard Crespi for the first time, was his idea of the evolution of the social brain. And Crespi had started to gain evidence that if we think of normalcy, there are extreme ends of the spectrum of evolution of the social brain. If you actually look at primates, the size of their social unit is directly related to the size of their brain. We spend a lot of time on social interactions and a lot of brain power. Bernie predicted that genetically these might be reciprocals, and at that point, only the 16p1.2 deletion had been found in association with autism. We went on to show it had microcephaly, while the duplication had macrocephaly, and then genome-wide association studies by Jonathan Sabat went to show that schizophrenia is caused by the duplication. Likewise, at 1Q21.1, the Baylor team, after the deletion was shown to be associated with schizophrenia, we showed it was associated also with head size. And have a recent paper coming out that's showing head size in the population is moderated by here. I think that reciprocal dupes and del enable you to change both ends of the penetrance spectrum that Thea was talking about. As she said, the point mutations, you can wipe some of them out different way, or the deletions, you can wipe them out easier. This way you can go either end of the spectrum by dupe or deletion. So important to evolution. And I can't get my psychiatrist colleagues to do it, and that's why I try to write in the journals. But this was described in 2008 at Baylor alone. We've diagnosed 253 families with this deletion or duplication. Conclusions in my last minute. NIH favors deletion. Replicative mechanisms favor duplication. Ectopic synapsis precedes ectopic crossing over. Triplication can form de novo by a double crossover at the LCR or from pre-existing duplications. triplications with non-recurrent breakpoints have a unique structure. Trip inversion do. Telomeres may be particularly susceptible to replicative mechanisms. Complex genomic arrangements can form by MMBR with template switches occurring both at homologous and microhomologous substrate sequences. And these CGR show many characteristics attributed to chromothripsis, or what we call chromoanasynthesis, and it's likely both mechanisms are operative in different cells. What are the issues important to this meeting? CNVs are important for disease, genomic disorders, and evolution. Do the current mutagenic assays, Bruce Ames test, measure CNV formation? Or are we missing this in all our environmental tests? Can we design the assay we all really need? Are we introducing compounds into our environment that induce CNV mutagenesis? I showed you from the multiple CNV families, it can be a rheostat, and we're looking for those genes right now. What is the evolutionary rheostat? Is it single nucleotide variation or copy number variation? Doesn't happen in a vacuum. From around the world, I am the most fortunate person, and my happy gene is not mutated, even though I'm a male. And have incredible postdocs and students and faculty colleagues. Thanks for listening. Happy to take questions.
Well, I'd like to thank Dr. Lupski for an amazing and insightful talk, and really full of information. So we'll take a couple of questions.
Hi, Jim. Intra-individual variation. So within an individual, you could look at cancer as an extreme.
Right.
And then you could actually say that it's unlikely that normal cells aren't generating CNVs at some clip. So can you comment on that? And,
I'd love to, John. Thanks for the opportunity. We actually published a paper in 2009 in Nature Genetics showing that the replicative mechanism can happen postzygotically, because that's when you go from one cell to 10 to the 14th to make us human being organisms. And how we saw it is we saw recurrence in a family of the same complex rearrangement. And to our surprise, when we got the breakpoint, we had PCR assay. We did PCR on the blood from mom and dad. We could find it in 1/1000th of mom's blood cell elements. So that mutagenic event happened after her 2-cell stage and before the split off of her germ cells from her blood elements. And that's why we could detect it in both of those. I think postzygotic mutagenesis is happening much greater than anybody really has an idea right now. And so cancer's the extreme. I think brain, lots of other places, lots of other diseases are going to be due to this.
Yeah, I was wondering if you could comment a little bit more about chromotripsis and being one catastrophic event, especially in cancer. How can you exclude all the selection that occurs?
Okay, if you're familiar with trying to get a paper through review itself, I think the guys at the Sanger did a pretty damn good job of ruling out all the obvious things. And they were left with this being the only explanation of their data. But I would say, read that paper. But I would also say that you're right. I mean, the paper was published not even a year ago. This field is woken up now to something they never thought about. And but you can make massive evolutionary changes by having such complex structures generated in one event.
So I'm wondering,
they do mathematical modeling to clearly show that there's not different concentrations of each portion of the rearrangement in the cell line, that you have the event happened once and everything in the tumor that's derived from it has that event.
So in the human disease studies, Do you find that CNVs also overlap regulatory regions in addition to the coding regions?
Okay, so it's remarkable, you know, we heard about the ENCODE project and the 30 papers that came out this month. We've been taught, at least I grew up with the idea of junk DNA, which I don't believe exists, and also gene deserts. Well, there's no such thing as a gene desert. It's a regulatory sea. So we are seeing CNVs that don't include genes cause very specific human disease phenotypes. I'll refer you to a paper coming out in Genome Research by one of my colleagues, Pavel Stankovic, which beautifully shows that in the case where he sees this happen, it's a link RNA that is taken out, that link RNA actually brings together the regulatory regions to specifically express during lung development, and it's a lung developmental disease that he finds it in, alveolar capillary dysplasia.
May Ho, Cincinnati. When you mentioned that, like in bone cancer, 20 to 30% of the cancer, 25% of the cancer cells actually have this big.
They only looked at 40, but it was pretty impressive.
Right. Well, but when you look at the primary site cancers, they have a much lower percentage, 2 to 3%. So, do you think it's a selection that means those cells that have this massive change of events can actually leave the primary site and end up in the bone? Or do you really think that there are two types, or there's a degree of change that will create some that will be latent, that we basically, for example, prostate cancer is dear to my heart. Yeah. People can live with prostate cancer and die with like prostate cancer, and for 20 years, and it really don't need to be treated.
I think these are great questions, and the field is such that it's so new that we don't have the answers yet. And I do think, as Thea once said up there, I don't have any data, so I'd rather not comment. We'll get some data.
And I'm really pleased that in some cases we only found one gene that is going to control our happiness. So.
Well, you don't know. It could be on X and just a dosage effect in females.
So Jim, sorry, I can't just ask one question, but one of your comments.
You can't because it's your second question.
Yeah. So I'll make a comment.
One more question.
Okay, so I guess the one thing that came up with the Sanger thing is you said, people are just aware. But there's a certain homocentricity about that, and that many human genetics, and I'm a geneticist and I'm in a department, don't know the history of the literature of studies done in Drosophila, E. coli, various other organisms.
It's multigenic inheritance,
so
right?
Though we are giving some of
poisoned us.
Yeah, though you are giving some of these things new names, some of them aren't that radically different from other types of DNA repair phenomenon that have been put forth. So could you speak to that? And I appreciate that you actually presented the background that shows that, but I think that needs to be reemphasized.
I tried to emphasize that, in one way, we called it microhomology mediated break-induced replication, but in fact, it was called that specifically because of the beautiful work on break-induced replication from Jim Haber's group, Lorraine Simonton, and their followers, Anna Malikova, Greg Ira, many other people. But the combination of using microhomology and long-distance template switching was in Lorraine's original Nature paper in 2007, but she never saw it beyond right where the breaks were happening, where we're seeing megabase size. Conceptually is, I think, enough different to give it a different name, but I'm happy to hear an alternative from you, John.